All right. Shall I go, Dario?
Yes, please. Thank you, Andy.
Okay. Well, it's my pleasure this afternoon to be introducing our speaker, Thi Nguyen. Thi's been a student at Davis for some time. You think of him as a triple threat. He got his bachelor's in V&E back in 2014. Did some work in industry after that. Did a harvest down in Argentina, but came back then and started a master's in ag and environmental chemistry, which he completed in 2017. But the research bug had definitely hit Thi, and he signed up, he re-upped for a PhD, also in ag and environmental chemistry, where he's been looking at the basic chemistry of oxidation, looking at various aspects of that, which I'll be discussing today. The other thing I think I wanted to mention is that Thi's a natural teacher and has been very involved in 123. Many of you here have had the privilege of having him as your instructor. And even through the pandemic, he's created the content for the online version of 123, which I think was very successful, given the constraints we had, but developed some really engaging materials and came up with some new ways to teach the course. So I'm looking forward to where Thi ends up, but to find that out, I know he's exploring a number of options, but I know his real love is teaching, and I hope that he ends up in a teaching position at some point. So with that introduction, I'll turn it over to Thi and let him begin his talk.
Alrighty, thank you, Andy, for that introduction, and thank you everyone for showing up today, especially those of you from outside the department and outside of the university. Even thanks for dropping by and listening to this ramble here. So, predicting wine aging, sort of a clickbaity title. We're nowhere near being able to predict wine aging, if by predicting you mean like being able to say that this wine will peak in 5 years or 10 years, or being able to prescribe a certain amount of oxygen to wine. Like we're not there yet. But we do have a better and better understanding of the microbial and chemical compositional parameters that control wine aging, which is what I'll talk about today. So just an overview of today's talk. I'll do a bit of intro about wine and wine oxidation research. Then I'll cover the mechanism of wine oxidation, the chemical mechanism of wine oxidation. This will be integral to understanding the 3 projects that I'll talk about here. So the first project has to do with microoxgenation and the role of yeast. The second project was the development and validation of a spectrophotometric method for iron speciation. And then the last project, well, I then used that method for iron speciation for this last project, which had to do with iron redox cycling and oxygen consumption. But I guess all of these are meaningless words for now. We'll know what they mean in a minute. So wine, obviously very important. It's a global multi-billion-dollar industry. I think it's made on every continent except for Antarctica, like 99% sure about that. So research in support of this global industry must also be global, right? So like what I'm doing here is just a small part of a much larger international research effort. Not only into wine, not only into wine chemistry, but even something as specific as wine oxidation research is getting attention. So just a couple examples of topics that people are investigating here. Microoxgenation, or MOX. This is a fairly recent technology by which oxygen is deliberately introduced into wine. And this is an attempt to simulate or even accelerate the aging process. And I say attempt because it's not always successful. It's been inconsistent, hence the need for research. And then another thing that people are looking into is premature oxidation, or premox. I guess that's pretty self-explanatory. So this phenomenon was first noticed in Burgundy, France, but it has since been observed worldwide. And this is when wines that are deemed to be age-worthy suddenly, inexplicably become undrinkable. I guess undrinkable is kind of dramatic. It's perfectly safe, but it doesn't taste as good as it should prematurely. So the overarching research question, for me at least, is why do different wines age differently? Or said another way, it would be why do different wines respond differently to oxygen? So the purpose of these research projects that I'm going to get into is to identify microbial and chemical parameters that can be, 1, measured to predict how wines will age, or 2, manipulated to alter the course of aging altogether. So a better understanding of such variables would allow industry members more control over the winemaking process, to achieve their desired style of wine and to give consumers the highest quality product possible. And a better understanding of these variables would also give consumers some indication of cellaring time or shelf life. Basically, like how long should I hold on to these wines, or how long can I hold on to these wines before they go bad? So let's dive into oxidation. What is oxidation? So in the context of wine, oxidation is a cascade of multiple chemical reactions that are triggered by oxygen. And these reactions will change wine's chemical composition and consequently change wine's sensory attributes like color, flavor, and mouthfeel being the main ones. Now, speaking from a purely chemical perspective, just to refresh your memory in case it's been a while since you've taken Gen Chem, oxidation is 1/2 of a redox reaction, or an oxidation reduction reaction, which is simply the transfer of electrons. So oxidation is the loss of electrons. When we say that wine is oxidized, it means that the compounds in wine are losing their electrons to oxygen. So if something is losing electrons, it has to be taken up by something else. That's reduction, the gain of electrons, and oxygen is being reduced. So there's always a give and take. So that is what's happening in the primary reactions here of the mechanism of wine oxidation. So what I've shown here on the left are phenols, or phenolic compounds. So these are the primary contributors to wine's color, flavor, and mouthfeel. There are other compounds, but for the sake of this talk, we'll just focus on phenols here. So phenols, when they're oxidized, they get converted into quinones, which is the form shown at the bottom there. So phenols are losing their electrons. Oxygen is what accepts those electrons from phenols. And doing so, oxygen is reduced into hydrogen peroxide. And then in the middle is iron. So iron here acts as a catalyst for this oxygen-phenol redox reaction. Without iron, without any metals, wine would not age. But we have no good legal way of removing iron from wine. And it's important to note here that oxygen cannot react directly with most organic compounds, phenols included. This is what's called a spin-forbidden reaction. Not going to get into it, but if it were to happen, I think the world would spontaneously burst into flame. Like oxygen does not react directly with organic compounds, thankfully. So iron is needed as a catalyst, and what it's doing is it shuttles electrons one at a time from phenols to oxygen, And in doing so, it redox cycles between iron 2 and iron 3. So iron 2 is what reacts with oxygen, gets converted to iron 3. Iron 3 then takes that electron from phenols, gets converted to iron 2, and so on and so forth. So iron, I like to think of this turnstile or revolving door for electrons. It's constantly passing electrons from phenols to oxygen. So those are the primary reactions. Then here are the secondary reactions. So once we've formed quinones, these quinones are electron deficient. They're electrophiles. So they will react with nucleophiles found in wine. And these nucleophiles include thiols and other phenols. And I'm not going to get into these reactions, but basically these reactions will further change wine's color, flavor, and mouthfeel. Then on the right, hydrogen peroxide will lead to the Fenton oxidation of ethanol. So the first step is the Fenton reaction. This Fenton reaction is also catalyzed by iron. And in the Fenton reaction, hydrogen peroxide decomposes into the hydroxyl radical. Now, this hydroxyl radical is incredibly reactive. It'll react with whatever it comes into contact with first. And in wine, the most likely target or candidate for oxidation is ethanol. So ethanol is oxidized by this hydroxyl radical into the hydroxyethyl radical. And then this radical is then converted to acetaldehyde. I'm kind of glossing over a couple steps, but that's basically it. It goes ethanol, then this hydroxyethyl radical intermediate, and then acetaldehyde. So acetaldehyde, this compound shown here, it's a volatile compound, so it'll contribute to the aroma of wine. It's actually the primary contributor to the oxidized aroma of aged wines or like mature wines or sherries, if you've ever had sherries. So at low concentrations, it confers just this sort of nondescript, pleasant fruitiness, just an added layer of complexity. But at high levels, people will describe it as smelling like rotten apples. It can smell metallic or nutty even. And pure acetaldehyde to me smells a lot like almonds, like some kind of almond pastry dipped in amaretto. And pure acetaldehyde is actually incredibly carcinogenic, and I have no business knowing what it smells like, but I do for science sake. So acetaldehyde, in addition to being a volatile compound, it also participates in additional reactions. Let's call them tertiary reactions with phenolic compounds. So an example of this is when acetaldehyde reacts with anthocyanins to form pyranoanthocyanins. Now, anthocyanins are the compounds normally responsible for red wine color. Anthocyanins are normally like a bright red-violet color, but pyranoanthocyanins are instead sort of a brick red to orange brown. So it's these reactions that explain why red wine color changes as it ages. Acetaldehyde can also bridge together flavonoids and you end up with these ethyl-bridged polymers. These little nubbins here are originally acetaldehyde, and you form these polymers, and this will lead to changes to wine's flavor and mouthfeel. So acetaldehyde is a marker for oxidation. It's responsible for the aged aroma and color changes to color and mouthfeel, but attempts to deliberately produce acetaldehyde in both a commercial and laboratory setting have had mixed success, which brings me finally to the first research project here. So microoxgenation, like I mentioned, is the controlled addition of oxygen to simulate or accelerate the aging process. And here's just a basic schematic of what's going on. You've got your tank of wine, there's an oxygen supply, a dosing system, not a dosing system, not my figure. So dosing system, and then oxygen is diffused as these little micro bubbles into the wine. So there are a couple commonly observed effects of MOX. There's pigment formation, which leads to more stable color. Like that red-violet color of young wines is not as stable as the brick red color of older wines. There's also improved fruitiness. I don't know if you actually increase the fruitiness or if you're simply decreasing the vegetal character, which then like unmasks the fruitiness and lets it shine. And then another effect is softened astringency. Now, what's been inconsistent is acetaldehyde production. And so there have been several others in the Waterhouse lab, my predecessors, that have looked at microox. So the first was Guomin Han. He was a visiting scholar from China who worked in the Waterhouse lab for a little while. And he found that acetaldehyde was produced only in unfiltered wine that contained yeast. And that's what this graph is showing here. We've got time on the x-axis in days, acetaldehyde on the y-axis, milligrams per liter. We can ignore the bottom three lines and just focus on the one that is increasing. It slopes upward. And you'll notice in the legend, it's that's a wine with yeast, and it's microoxed. So yeah, acetaldehyde requires yeast and oxygen. And then Charlie Henshin, who was a master's student in viticulture and enology here at Davis, did a similar experiment with some unfiltered wine. And he noticed that acetaldehyde increased upon the depletion of SO2, and this coincided with a bloom of yeast. So that's what these graphs are showing here. Days on the x-axis. Up top, we have yeast cell counts and colony-forming units per mil. So SO2 is depleted at some point between 30 and 40 days. And these, this purple and green lines, there's an increase in viable yeast or culturable yeast. So these yeast were probably dormant for most of the experiment, viable but non-culturable, and suddenly they bloomed with the depletion of SO2. And you can see that this coincided with an increase in acetaldehyde. Again, the purple and green lines here. So yeast and oxygen were what was needed to produce acetaldehyde, but it still wasn't consistent. Like acetaldehyde was produced in the wines with yeast and oxygen, but not all wines with yeast and oxygen produced acetaldehyde, meaning that there was at least one other variable that was unaccounted for. So Dr. Waterhouse and I looked at both Guomin's data and Charlie's data, and we found the common thread to be residual sugar. So acetaldehyde production with MOX requires viable yeast and residual sugar. So it would seem to be the case that the three ingredients for acetaldehyde production were oxygen, yeast, and sugar. So what I did to confirm this was I fermented some synthetic must. This is basically an artificial grape juice just to have a nice controlled, well-defined chemical environment. So this must was fermented to dryness using Saccharomyces cerevisiae strain EC1118. Experimental treatments here. I either added glucose at 3 grams per liter or I did not add it. The wine was either oxygenated or not oxygenated. The cell density immediately after fermentation was on the order of 10 to the 8th colony-forming units per mil. So this was at the end of fermentation, but the start of the experiment. So initial cell density. Then I quantified acetaldehyde by HPLC DAD before and after 10 days at room temperature, basically. And this is what was found. So x-axis, the different treatment combinations, y-axis, acetaldehyde. And the dotted line shows the initial acetaldehyde concentration at 70.4 milligrams per liter. And you can see here that acetaldehyde increased with both glucose and oxygen. So that's the first bar in GLU-OX there, and it decreased in all other cases. So my best guess as to why this is happening is that oxygen precludes the need for NAD+ regeneration by ethanol formation. What do I mean by that here? So we can take a look at the metabolic pathway for yeast. Shown here are both the fermentative and respiratory pathway of yeast metabolism. So in red is the fermentative pathway. So normally, like under normal winemaking conditions, glucose gets converted into pyruvate via glycolysis. And this process uses the cofactor NAD+, reduces it to NADH, right? And then pyruvate gets taken to acetaldehyde. And then the final step of alcoholic fermentation is when acetaldehyde gets converted into ethanol. This process of fermentation recycles NADH back into NAD+, needed for continued glucose consumption. Now, if you have oxygen, I think what's happening is that the electron transport chain is active. So you have an alternative means of recycling NADH, right? So rather than converting acetaldehyde into ethanol, that NADH can instead feed into the electron transport chain, and you no longer need to produce acetaldehyde. So the yeast allows no longer need to produce ethanol. The yeast can just let acetaldehyde accumulate there. That's what I think is going on. You might want to get an actual microbiologist to look into this in the future. So there's still a question then of why chemical acetaldehyde production is not occurring, right? Like without yeast, without residual sugar, why isn't the mechanism of chemical acetaldehyde production working as it should? So during my time here at Davis, I've had multiple conversations with Dr. Roger Bolton, who has suggested that iron 3 is locked up by oxygen. I Like if you have enough oxygen there, iron 3 is sort of frozen in place and it doesn't redox cycle as it should. So this idea has been echoed by Danilovich in 2016. That's what this figure is suggesting here. It seems to be suggesting that the oxidation of iron 2 to iron 3 by oxygen is fast, whereas the complementary process of reduction of iron 3 back to iron 2 by phenols is slow. So this hypothesis now is that oxidation is limited by the ability of phenols to recycle iron 3 back to iron 2. Like the supply of iron 2 is not enough to meet with the demand for iron 2 by oxygen. So to test this hypothesis, I first needed a method of doing iron speciation, like a quick and easy method that I can do rapidly, just to get a good look at the kinetics. So spectrophotometry is quick and easy. I went with that. And just a definition of iron speciation. It's a means of distinguishing and quantifying the two oxidation states of iron separately. Rather than measuring total iron, I have to be able to measure at least one of them, right, and distinguish it from the other species. So it's this method that I will then go on to determine the rate of reduction of iron 3 to iron 2. So the reaction on the left here, by measuring the formation of iron 2. So there have been previous methods published, spectrophotometric methods for iron speciation, and these are the commonly used complexing agents. So those of you that have taken VEN290 will remember ferrozine. Here are a couple others. You'll notice this, I don't know if you can call it a functional group, this structural feature here with nitrogen atoms. It's this feature that allows complexation with iron. So it takes 3 of each of these molecules and 1 iron ion to form a colored complex. So it takes this octahedral geometry here. And it's this color formation that allows us to measure iron. Now the issue with all these complexing agents is that they are iron 2 selective. So by stabilizing the reduced form of iron, iron 2, you're increasing the reduction potential of iron. You're making it easier for iron to be reduced. Now that is problematic because during analysis, preferential stabilization of iron 2 causes reduction to occur during your analysis. Like you're trying to measure iron 2, but you're forming iron 2 as you're trying to measure it. So you'd get inaccurate results. And that's what this figure is showing here. Time on the x-axis in minutes, and then the increase in iron 2 on the y-axis here. So you can see the line that slopes upward is when ferrozine is used alone. You can see that iron 2 is being produced as you're trying to measure it over the course of 6 minutes. Now what I did to modify this was add an additional complexing agent. So the relatively flat line at the bottom is ferrozine plus EDTA, a second complexing agent. This allows stable measurements to take place. So the use of ferrozine and EDTA captures a stable snapshot of iron species. And EDTA is ethylene diamine tetraacetic acid. So how this method works. So ferrozine, like I said, it's iron 2 selective. It'll bind iron 2 and it forms a magenta colored complex that's measurable at 562 nanometers. So that's for iron 2. EDTA, on the other hand, is iron 3 selective. So it'll bind iron 3 and it actually forms a colorless complex that doesn't interfere at all with measurements of iron 2. So what I've done essentially is use 2 complexing agents, one for iron 2, one for iron 3, pull apart the 2 iron species, lock them in place, and measure just iron 2. So that's how this method works, and this is the method that I will be using for the 3rd project. Or I have used for the 3rd project. So yeah, I'm measuring the rate of this reaction here, the reduction of iron. Now bear in mind that the reduction of iron is the same thing as the oxidation of phenols, right? So phenols are being oxidized by iron 3, getting converted into quinones. But here I'm keeping track of the iron 2 concentration, the formation of iron 2 from iron 3. So a couple questions that I'm asking here with this project. How does chemical composition affect the rate of this reaction? And can this reaction keep up with demand from oxygen? Like is supply enough to keep up with demand. So this experiment, did it in model wine, again, for a nice controlled chemical environment. So this is 12% ethanol by volume, 53.3 millimolar tartaric acid. That's the main acid found in grapes and wine. This is equivalent to 8 grams per liter. Then iron 3 was included at 0.1 millimolar. This is 5.5 milligrams per liter. This is the global average iron concentration in wine. Then I added 1 millimolar of 4-methylcatechol. So this is just a model phenolic compound. It doesn't actually exist in wine. That's because getting pure, like real phenolic compounds is way too expensive. But 1 millimolar of 4-methylcatechol is about equivalent to 290 milligrams per liter of catechin. So this is about the concentration of phenolics in white wine. Also included was 1 millimolar benzene sulfinic acid. So this is a nucleophile that's required to consume this quinone and pull the reaction forward. And measuring the rate of iron reduction, this was done in anoxic conditions. So I got dissolved oxygen levels down to practically 0. And this is because I didn't want any interference from the reaction on the right, like as I'm measuring the reaction on the left there. So I looked at several different chemical parameters here, but I just want to focus today on the effects of pH because I think these are the most interesting. So time on the x-axis in minutes. Keep that in mind that the time scale is in minutes. That'll be important later. Y-axis shows the formation of iron 2 in millimolar. And you can see that this reaction, the reduction of iron or the oxidation of phenols, actually slows down as you increase in pH. So this was a little unexpected. Like I always think that, oh, wine oxidizes faster at higher pH, but this reaction actually slows. So you can see it's pretty fast at pH 3, slower at pH 3.5, and even slower at pH 4. Now, why is this happening? So my best guess is that this has to do with complexation of iron 3 by tartrate. So tartrate, like I mentioned, is the main acid in wine, and it was what was used in this model system. So tartrate can form these complexes around iron 3. So what's shown here up top is a monomeric iron 3 tartrate complex. Down below is a dimeric complex, and there's also been trimeric complexes observed as well. So tartrate prefers iron 3 over iron 2. So it stabilizes the oxidized form of iron 3. So this decreases the reduction potential of iron. Preferential stabilization of the oxidized iron 3 form makes it harder to reduce iron. And what I think is going on is tartrate competes for the coordination sites of iron 3. So basically, like you've got tartrate surrounding these iron molecules, blocking phenols from reacting with iron 3. That's what I think is going on. And this effect is pH dependent because complexation requires deprotonation. So you'll notice that it's not tartaric acid that is complexing with iron 3, it's tartrate, the deprotonated form. And this occurs more readily at higher pH, so you've got more abundant complexation as pH increases. So that would explain the slowing of this reaction as pH increases, I think. Now, I took the same model wine system, saturated them with air, and this brings the oxygen level up to a maximum of about 7 milligrams per liter. And I let these wines consume that oxygen. So here I'm tracking the rate of oxygen consumption. And for all the same reasons, this reaction speeds up as you increase in pH. So this again has to do with tartrate preferring the oxidized form of iron 3 over iron 2. So you can see here this graph, this time scale now is on hours. Okay, so that's the x-axis and then the oxygen consumption on the y-axis in milligrams per liter. And you can see it's slow at pH 3, faster at pH 3.5, and even faster at pH 4. So the opposite effect of pH is observed here for oxygen consumption. Now, both iron reduction and oxygen consumption followed pseudo-first order kinetics. And from this data here, I was able to calculate pseudo-first order rate constants. And that's a key detail that I think is worth just, I'm gonna review the definition of pseudo-first order. just to jog your memory in case it's been a while since you've taken Gen Chem. So pseudo-first order kinetics, this happens when the rate of a normally second order reaction appears to be first order with respect to one reactant when the other reactant is in significant excess. So normally a second order reaction depends on the concentration of two reactants. But when one is in excess, the reaction looks like it really depends on the one that is limiting, the limiting reactant. So this makes sense for iron 3 reduction. So the process of iron 3 reduction was pseudo-first order with respect to iron. So iron was included at 0.1 millimolar, and its reactant, 4-methylcatechol, was at 1 millimolar, so a 10-fold excess relative to iron. So that makes sense that this reaction is pseudo-first order with respect to iron. Now, oxygen consumption, this was kind of weird. It was pseudo-first order with respect to oxygen, which doesn't make sense too much. So oxygen was at 7 milligrams per liter, so this is 0.2 millimolar. But its reactant, the iron, was present at only 0.1 millimolar, which is half the concentration of oxygen. So it's not even in excess, right? But yet oxygen consumption followed pseudo-first order kinetics. Now, why might this be? So here are the calculated pseudo-first order rate constants at 3 different pHs, 3 to 4. Iron reduction and oxygen consumption. You'll notice that these rate constants, so iron is on the order of 10 to the minus 2, 10 to the minus 3, whereas oxygen consumption is 10 to the minus 5, minus 4. So more than an order of magnitude, like at least an order of magnitude greater than oxygen consumption. So it turns out here that this reaction on the left, the one that I've shown in bold, is at least 10 times faster than the reaction on the right, which is shown with the dotted line there. So yeah, it seems like iron 3 is not really locked up as iron 3 at all. So there have been a couple studies in which copper is shown to speed up oxygen consumption. So I added copper into these wines to see whether the rate of oxygen consumption could overtake the rate of iron 3 reduction. So I repeated the experiment with some copper. This is, I think this is just above the legal limit for copper. So 0.01 millimolar. So this is a pretty extreme scenario, right? Just above the legal limit. And it didn't have much of an effect on iron 3 reduction, but it did increase the rate of oxygen consumption significantly. But still, like all these rate constants were an order of magnitude less than those of iron reduction. So this reaction on the left is still going much, much faster than the reaction on the right. So I would come to this conclusion here that the oxidation of iron 2 by oxygen, again, that's the reaction on the right, is the rate determining step for the wine oxidation pathway. So just as a couple follow-up conclusions here, iron, again, is not locked up as iron 3, even when oxygen is in excess. So what this means then is that wine oxidation is not limited by the recycling of iron, but by oxygen ingress. Like wine is pretty much oxidizing as fast as it can. It's able to consume that oxygen that you give it. These conclusions aren't without precedent here. There have been a couple studies that have attempted to correlate wine compositional parameters, things like phenolic content and composition, to oxygen consumption rates. And what these studies found is that for most compositional parameters, there was either a very poor, weak positive correlation, no correlation at all, or even negatively correlated. With the exception, so the only 2 compositional parameters that were positively correlated to the rate of oxygen consumption were pH and copper. So it's as though the rest of wine composition has no bearing whatsoever on oxygen consumption rates. And a couple other observations to support this. So for wines in storage, iron 2, iron 3 ratios are pretty high. So most of the iron is present as iron 2. And this aligns with low oxygen levels found in wine. So again, oxygen is never really in excess, like it's being consumed and kept at low levels. So, I mean, we still have this question here, why is chemical acetaldehyde production not occurring? Right, so we can do away with this notion of lockup of iron 3. We know that the reaction on the left is faster than the reaction on the right. And I think this question is not even asked properly. I think there's a better way to ask this question. So rather than asking, why is chemical acetaldehyde production not occurring, we should instead be asking, why does acetaldehyde not accumulate? So I think that the reactions that lead up to acetaldehyde are still active, but there are a couple like hiccups along the way that prevent this, prevent acetaldehyde from accumulating. So the first possibility is that the hydroxyethyl radical intermediate, remember this is the intermediate that is between ethanol and acetaldehyde. Okay. So it's possible that this intermediate is quenched by phenolic compounds, mainly hydroxycinnamic acids found in wine. And this is what Nick Gislason showed in 2011 here. This is what this figure is showing. So this compound here is the hydroxyethyl radical, and normally this gets converted into acetaldehyde. But it can be intercepted by hydroxycinnamic acids, and through a series of reactions produces this, I guess it's an alcohol. This alcohol and the CO2 leaves. So rather than letting acetaldehyde form, it gets diverted here by hydroxycinnamic acids. Now, another possibility that acetaldehyde is being produced, but it's being consumed more quickly than it's being produced. So remember I mentioned these pyranoanthocyanins. So anthocyanins can react with acetaldehyde and produce these pigments. There's also flavonoids reacting with acetaldehyde to produce these ethyl-bridged polymers, right? And Bueno et al. In 2018 coined the term aldehyde reactive polyphenols, or ARPs. So it's these phenolic compounds that are sort of sequestering acetaldehyde, or like tucking it away and keeping it hidden and not allowing it to accumulate. So yeah, two possibilities there as to why acetaldehyde is not accumulating. Definitely worth investigating. So some additional research questions for the future. So this first question I'm actually looking into right now. This is my final project before graduating. So I'm interested in seeing how the wine acids, their complexation of iron by wine acids affects the Fenton reaction. So we've seen how tartrate at different pH can influence the rate of iron redox cycling. Right now, the Fenton reaction is also catalyzed by iron, and it's my hypothesis that not only tartrate, but other wine acids, malate, lactate, acetate, citrate, at different pHs will have differential effects on the Fenton reaction. So that's what's going on right now. That's what I'm doing at the moment. And then some additional research questions. So rather than asking how fast is oxygen consumed, like rather than looking at the rate of oxygen consumption, I would focus on the capacity for oxygen consumption. So rather than asking how fast, we should ask how much oxygen can be consumed until some chemical or sensory threshold is reached. And what I have in mind is asking the question, how much oxygen can be consumed until radicals can no longer be intercepted, right? Those hydroxycinnamic acids that are quenching the hydroxyethyl radical. When is their ability to quench radicals depleted? Or how much oxygen can be consumed until acetaldehyde can no longer be sequestered? Right? Like when do you run out of ARPs that are tucking away acetaldehyde? And so yeah, this is a worthwhile research question, I think. If we're able to be able to predict wine aging, we would need to measure the radical quenching capacity of wine or the acetaldehyde consuming capacity of wine. I'll leave that to someone who is bright-eyed and bushy-tailed to look into. And with that, I'd like to thank you for sitting through this talk. A couple of acknowledgements here. The ARCS Foundation, that's the, what is that? The Achievement Rewards for College Scientists Foundation. They funded this research. Also the American Vineyard Foundation, supported me, gave me rent and money for food and things like that. The Department of Viticulture and Enology has been my home for the past decade. That's really gross to say. Yeah, it's been a decade in the V&E department. And then the AgChem department, also AgChem graduate group, also second home. Members of the Waterhouse Lab, of course, Andy Waterhouse, who's gone. There's another meeting to attend to. My principal investigator, my advisor and mentor, James Campbell, a lab mate. I don't think he's here. He's on a beach somewhere, drinking, no doubt. and Pat Howe, lab manager and my co-instructor for VEN290 fall quarter. Have all been wonderful support. And I also want to just thank my friends and family who have kept me sane for the last 6 years in grad school. And with that, I'd like to open it up for questions.
Excellent. Thank you very much for the seminar. And if there are questions, either the chat or unmute yourself, and we have 10 minutes. So yeah, Michael, please go ahead.
Hi, great job. Really interesting. Did a lot of good work. I had a question about the formation of acetaldehyde by yeast, because when they're active, right, when they're consuming oxygen, they also have a second enzyme, aldehyde dehydrogenase. So yeast can actually live on ethanol, right?
Yeah.
And it's those two enzymes, the ethanol, alcohol dehydrogenase followed by aldehyde dehydrogenase, and they produce acetic acid. So is there a correlation between acetaldehyde production and acetic acid production? Is it just that a certain part of the acetaldehyde goes out in solution and doesn't get consumed by the aldehyde dehydrogenase?
So yeah, there are those enzymes, but I think under normal winemaking conditions, the respiratory pathway, those enzymes aren't active. Like you don't really have ethanol being reverted back to acetaldehyde, back all the way to acetic acid.
But that's what I assumed, that the yeast, maybe I missed that. So I assumed that the yeast, when you microoxygenate, the yeast were necessary because of the ethanol oxidation. Because you were showing that the oxygen goes into the respiratory chain, right? The respiratory chain begins with NADH, and then NADH would have to come from alcohol dehydrogenase, right?
Yeah, so I mean, yeah, there are some yeast strains used in sherry production that do use respiratory metabolism to convert ethanol back into acetaldehyde, but I think it would be weird for yeast that had just left a fermentative environment to suddenly change their enzymatic machinery and switch over to an entirely different pathway there.
So there's no correlation between production of acetic acid and production of acetaldehyde in those microoxgenation experiments?
Well, I'm not entirely sure, not that I know of, yeah. Finally got back to this pathway and the questions over. Yeah.
So there are many messages of congratulations in the chat. No questions.
No question.
Hey, Thi, I got a quick question for you.
Yes.
Yeah.
So this might be a little contentious, but you've added oxygen with low oxygen and high oxygen. Have you looked at redox potential as a function of that oxygen level and its ability to influence the kinetics of these reactions?
No, I've not used an ORP probe ever. Yeah.
Would you like to?
Maybe if I do a postdoc that involves this.
Okay, fair enough. I'm just curious. It's really interesting work, and I know that Bob Coleman and I have both been looking at some of this stuff and would be fascinated to understand the effect of this chemical system you've worked out. And I love the sort of EDTA and the other molecule, so you can kind of freeze both iron species, but then just looking at the influence of redox potential and maybe adding oxygen in a way to influence that redox potential, and then looking at the particular kinetics of those reactions or which way it shifts the equilibrium.
Yeah, definitely something to look into.
Cool, great talk, amazing work.
Thank you.
Hi, Thi, congratulations. They're so proud of you.
Hey, thanks, Mary.
Love you, Thi.
Any more questions?
See, I did, but now I can't remember what it was.
Okay, everyone. Then, Thi, congratulations, and see you around, I guess.
Yeah.
For a while.
Very good.
Thank you, everyone. Bye-bye.
Thanks,
Take care.
y'all.